Hello everyone. Today we are going to explore an essential and very practical topic in histology, special staining methods used to demonstrate specific tissues. Have you ever wondered how nearly transparent biological tissues become so vividly distinguishable under the microscope? This transformation is made possible by staining techniques, which provide contrast and help identify specific cellular components with precision. Let us begin by understanding the basic concept. Biological tissues in their natural state have very little contrast under the microscope. Staining enhances visibility and highlights particular features of interest. When the chemical basis of staining reactions is well understood, we call this histochemistry. In this guide, we will look closely at three core methods: hematoxylin and eosin, periodic acid Schiff, and Masson trichrome, with labeled callouts and interpretation tips. Stains interact with tissue components through chemical affinity, charge interactions, or selective oxidation and reduction reactions to provide diagnostic contrast. Basic dyes bind acidic structures such as nuclei, while acidic dyes stain basic components like cytoplasm and extracellular proteins. Histochemistry extends this idea by targeting specific chemical groups or macromolecules, revealing carbohydrates, connective tissue, or microorganisms selectively. Good staining practice relies on consistent fixation, proper sectioning, and controlled timing to ensure reproducible color development and minimal artifacts. Interpretation depends on knowing which structures each stain highlights and on comparing stain fields with appropriate controls. We will now examine individual methods, starting with hematoxylin and eosin, the routine foundation for morphology and pathology assessment. Hematoxylin stains nuclei blue or purple by binding DNA. Eosin counterstains cytoplasm and extracellular matrix in shades of pink. H&E provides excellent overall tissue architecture, nuclear detail, and general pathology screening, making it the most commonly used stain. Pay attention to nuclear chromatin pattern, cell size, and tissue organization. These features guide whether special stains are needed for further diagnosis. Common H&E artifacts include overstaining uneven sections or poor fixation. Recognize artifacts to avoid misinterpretation. H&E often tells you which special stain to apply. For example, suspected carbohydrate accumulations suggest a PAS stain next. Now we will look at the periodic acid Schiff reaction, used to demonstrate carbohydrates and mucopolysaccharides. The PAS reaction oxidizes carbohydrates to aldehydes, which react with Schiff reagent to yield a magenta color highlighting glycogen and mucins. PAS is useful for identifying fungal cell walls, basement membranes, and stored carbohydrates. Controls with diastase can confirm glycogen presence. Masson trichrome differentiates muscle, collagen, and fibrin by using multiple dyes producing red muscle and blue or green collagen depending on protocol. Trichrome stains are valuable for assessing fibrosis and extracellular matrix expansion. Quantification tools often measure the stained collagen area. When interpreting special stains, always compare to H&E and use appropriate positive and negative controls to validate staining specificity. Next, we cover practical workflow tips for laboratory preparation, staining consistency, and avoiding common pitfalls during special staining procedures. Start with high-quality fixation and consistent section thickness. Variability here is the major source of inconsistent stain results. Standardize reagent preparation and incubation times. Small deviations in concentration or timing often produce large color differences. Use known positive controls for each stain and include negative controls or enzyme treatments when appropriate to confirm specificity. Document staining protocols and batch records to enable reproducibility. Good records speed troubleshooting and maintain quality assurance. When uncertain, consult reference atlases and experienced colleagues. Correlate histology with clinical information for accurate interpretation. Summary: staining transforms transparent tissues, H&E shows architecture, PAS reveals carbohydrates, and Masson highlights collagen. Controls and technique ensure reliable results. And that wraps up today's topic. If this explanation helped you understand the concept more clearly, make sure to like the video and subscribe to this channel for more simplified lab technology and radiology lessons powered by AI. We break down complex subjects in hematology, biochemistry, microbiology, pathology, X-ray, MRI, and diagnostic imaging, step by step, so you don't just memorize, you truly understand. If you have any questions or want a specific topic covered, drop it in the comments below. Until the next video, keep learning smart, keep thinking critically, and keep mastering diagnostics.